Hi guys, time to start on lab number 7, the staining of special structures. Now we talked about staining the bacterial cell walls and all of that, the gram stain. We've talked about simple stains. Here's our third type of stain. These are for specific structures, and to enumerate them, we're talking about spores, bacterial capsules, and flagella. These guys take special techniques in order to have them take up our color or make them visible. And we'll talk about that upcoming right now. So as far as the spores, these guys, we remember that the bacteria, if they're in a hostile condition, situation, environment, where it's unlikely that they're going to be able to survive, in order to propagate their species and all this good stuff, what they'll do is they'll actually go through almost like a binary fission, only it's not going to be walled off. And they're going to, it's called sporulation. And these guys are going to put themselves inside this very thick, impervious coat. When I say impervious, I mean that very little can get through there to destroy the bacterial DNA and the rest of the structures that the bacteria needs to come back. So the spore is almost like a little time capsule. When it opens, or when the conditions are right, the bacteria can go from the spore into becoming what we call the vegetative cells, the normal cells that we think of. Some of the more pathogenic bacteria form spores, these endospores. The bacillus. Now, a lot of the bacillus are fairly innocuous. They'll give you like a gastroenteritis, Bacillus cereus. But there's one species of that I'm sure everyone's heard of. It's Bacillus anthracis, which is anthrax. Very, very dangerous. Very, very lethal disease. The Clostridium, I know at least, without thinking hard, 4 species that are really nasty. Perfringens causes gangrene. Tetani causes tetanus. Let me think. C. difficile, Clostridium difficile causes a very nasty diarrhea. It can be life-threatening, as a matter of fact. And we've got one more, C. tetanus, which is lockjaw. I'll think of it, and I don't know why I never can think of that last one, but I'll think of it here in a few minutes. But in any case, this is why the endospores are very important. These guys are not, they're almost like in suspended animation. They are not metabolizing. In other words, they're really not doing anything. They're basically in suspended animation, heat, drying, no food, temperature extremes, and even chemical agents like antibiotics and even ionizing radiation cannot penetrate that spore. I mean, there's ways of destroying these guys, and they take special techniques. We talked about that in the lecture. But they're really a humongous pathogenic virulence factor. So when these guys form, we already said when the environment is hostile, and once the endospore forms, the cell itself will disintegrate, and the endospores will just stay dormant until things turn favorable. So we need to be able to identify endospores. Now, endospores don't take stains so well. So when we do this, we'll go through the procedure in just a minute. We actually have to use steam to help drive the stain in. capsules. Whoops. You undo that. There we go. Capsules. Excuse me. Capsules are another virulence factor that can make the difference. And remember, I've told you about there are two species or two types of Streptococcus pneumoniae. The pneumococcus can come either in a encapsulated, one that has a capsule, or one that doesn't. If it has a capsule, it's going to be much more difficult for our white cells to grab it and to take it in for phagocytosis. So it's a very important virulence factor, and we need to know this. These guys are going to be made of polysaccharide, polypeptide, going to form this impenetrable, very thick coat. Now, it can be a capsule when it's nice, round, and even, and irregularly shaped is going to be called a slime layer, but it's still a capsule, and it's more loosely bound to the bacteria. And some of these bacteria, if you watch some of the videos at the bottom of the lab, and you really should, can have capsules that are fairly large, much larger than the bacteria itself. Anyway, so this is something that is genetically determined. You remember we talked about the two types of pneumonia. Most polysaccharides, and this is the technique. We don't stain the capsule. We put stain in the bacteria and the background. And so what's left, you've got this colored bacteria and the background is colored, and you've got this area that's not colored around the bacteria. That's the capsule. And some protocols, they do it a little bit different. As a matter of fact, I think the lady that was, it was a lady doing the capsule stain had an interesting take on it, but we'll just go through it the way that we've got in our lab and won't go looking for all the possible ways of doing something. Flagella are those little hair-like structures that enable the bacteria to move. If the bacteria can move, that is an incredibly powerful virulence factor. It means these guys can move around and get away from our white cells and also colonize in places that really are really sensitive to those kind of infections. They can have one flagella at either end. They can have two flagellas at either end, in which case they're called polar. Or they can have flagella that goes all around their bodies or more than just on the polar. Those are the paratrichous and flagella. Thin protein-based structures project out. This is something that we need to remember. Very fragile, and it's not going to be visible with a light microscope. It is hair thin, thinner than a human hair, okay? So these guys are very fragile, and they're very, very small, even compared to a bacteria that's small. And bacteria are tiny under the microscope, even on maximum magnification. So we stain these guys. What we're actually doing is not coloring the flagella. What we're doing is actually coating it, and more coats. We're making it larger in diameter. So this is kind of an unusual stain for us, that it's not so much about color for contrast as it is just making it bigger so we can see it. And again, flagella, paratrichous or polar, absence or presence is very important in determining this. And when we do the unknown bacteria, all three of these are going to be important. The absence or presence of all three of these can help you narrow down which bacteria that you're actually looking at. Normally we do this with B. cereus or B. megaterium. They're fairly non-pathogenic. Here's what we use. We use a stain called malachite green. What we do is we have a beaker, we put the slide on it, we've got the bacteria on there, and we're going to use the malachite green with boiling water underneath in the beaker. The beaker's sitting on a hot plate, and the steam comes up. We put a, let me just wait and go through this when we get to the procedure. I can tell you the whole thing, but I think it's better not to. Capsule stain, nigrosin, which is for the background, methylene blue, which is for the actual bacteria. The flagella stain is called Leifson's flagella stain. And again, this guy is more of a coating thing than an actual coloring agent. Okay, guys, let's go ahead and go through the procedures. And again, I strongly advise you to watch those videos. If you take the links, they're really good and they'll help you guys. Okay, so we take our heat fix smear for the spore stain. We're going to cover it with a small paper towel. We take and we tear a little square off a paper towel. We don't want it to hang over the edges of the slide. We're going to put about, well, actually I'd put probably about 3 inches of water, because sometimes it takes about 20 minutes of boiling. It says 5 to 10, but in reality it's taken longer. We put the slide over the top of the beaker so it sits on the top. We boil the water and then the steam goes up, and then on that paper towel that's covering the bacteria, that's where we're going to actually put our malachite green, the spore stain, and we're going to keep watch on that and make sure that the paper towel doesn't dry out. We don't want to put so much that it floods over the sides, but we want to keep that paper towel moist with the malachite green. And with that steam, it's going to help to force that green stain into the endospores. We remove the slide after about, I'd say, honestly, probably about 15 to 20 minutes. Turn it off. We get rid of the paper. We're going to really gently wash it just a very short time. What we're basically wanting to do is to get all that extra green off the slide. It should be a very light tint of green, if anything. Now, this is where we do our counter stain. We're going to use safranin for 1 minute. Then we're going to rinse and shake off the excess. The green stain, the malachite green, was actually washed out of the vegetative cells when we did this washing in about for the 20 seconds. And so now when we put the counter stain in, we get the pink red color of safranin. Gently blot. I hope you guys have picked up that on blotting you don't want to rub it because you wipe off the bacteria. You just got to gently pat it. And then from that point you can put it under the microscope. You should see the pink cells and the green endospores. And the green endospores, usually a lot of them are going to be inside the pink cells. Depends. Capsule stain. We're going to heat fix this Enterobacter aeruginans, Serratia marcescens, either one. We're going to stain the smear for 1 minute with nigrosin. Wash the stain off with water. Nigrosin is a background stain, guys. It's going to be attracted to the glass, not to the bacteria. Then we're going to stain with 1 minute with methylene blue, which is going to be attracted to the bacteria. when we wash it off and then we blot gently. When we look at that under the oil immersion lens, we should see the blue, black, purple bacterial cell, and we should have a dark black ground, almost black, and then there should be a cleared space around these guys, and that will be the capsule. The flagella stain, this one is different than anything you've ever done before. We clean the glass first off with an alcohol wipe, and then we flame it. And right after we flame it, we let the slide cool. Then we put Proteus or E. coli, both of those have flagella. We allow it to air dry. This is the only one we definitely do not heat fix. If we heat fix it, we'll destroy the flagella. We're supposed to draw a rectangle under the dried smear on the opposite side, and we place it on a staining rack, or we put it over the sink is what we actually do. Now here we're going to actually flood the slide with that Leifson flagella stain and keep it within the rectangle. We're going to let it incubate at room temperature for 7 to 15 minutes. And again, the best time depends on the room temperature, and it can be longer. And it also depends on, well, it depends on the humidity and such. And what we're looking for is a golden film. There, you can actually see it on top of the stain. It kind of turns like gold foil, and it's going to develop on there, and we're going to see it through the sample. What we're going to do, and I disagree with this, I think what you need to do is not use tap water. I think you just need, like they say in the videos, just turn it at like a 45-degree angle and let some of the excess run off. And then you look at it and you should be able to see the bacterial bodies and the stains, and and the flagella. This is a really good site, guys. Now, hopefully you guys know the control click. You hold down the control button and then click with your mouse. Go to these sites and check those out. They're very good. All right, guys, that's got everything for lab number 7. And I will see you on the serial dilution lab shortly.